All right, so this is chapter 4, the functional anatomy of prokaryotic and eukaryotic cells. But this section we're just going to cover the eukaryotic cells. So first we're going to take a moment to compare the eukaryotic and the prokaryotic cell, because there are some major differences and some major similarities that you need to be aware of. So comparing prokaryotic and eukaryotic cells, the prokaryote comes from the Greek words for pre-nucleus, and the eukaryote comes from the Greek words for true nucleus. And so comparing these two, we have the prokaryote is going to have one circular chromosome, not in a membrane. There's not going to be any histones. There's no organelles. The bacteria are going to have peptidoglycan walls. Archaea are going to have pseudomurin walls, and they're going to divide by binary fission. In eukaryotes, we're going to have paired chromosomes in a nuclear membrane. We're going to have histones. We're going to have organelles. We're going to have polysaccharide cell walls when they are present, and they're also going to divide by mitosis. So here's your check your understanding questions. What are the main differences? These are really good questions to ask. Comparing and contrasting different things is always a great thing to ask. So this here is an idealized eukaryotic cells. And so there's a couple of things that are in plant cells only. And so the things that we only see in plant cells are the vacuole, the cell walls, and the chloroplasts. The cell wall is going to help to give it structure. We won't learn too much more about cell walls for plants in this class because that's just not the subject matter. But they're very important to the structure and function of plants. The chloroplast we will learn some more about because we will see it in our microscopic algae. And so the chloroplast really creates that the energy or the fuel for the cell to burn in that energy. And the vacuole is going to have and store water inside the plant cell. Some things we only see in animal cells, which is going to be down here in the bottom. We're going to see the centrosome, so centrioles and the paracentriolar material, the lysosome, the basal body, and the flagellum. For these things, you're only going to see these in animal cells. In both plant and animal cells, we're going to see a lot of things. That's over here. We're going to see the peroxisome, the nucleus, the nucleolus, the rough endoplasmic reticulum, the smooth endoplasmic reticulum, microtubules, microfilaments, the mitochondrion. This is where you take the fuel and you turn it into ATP energy. We're going to see the plasma membrane in both plants and animals. We're going to see ribosomes, the cytoplasm, and the Golgi complex in both of them. So eukaryotic cells, like I said, you can see some of these typical structures. And so this is a transmission electron micrograph of a plant cell, and you can see the chloroplast, and you can see the vacuole as well as the nucleus. So our learning objective for this next section is to learn about flagella and cilia. And so flagella and cilia are projections used for locomotion or moving substances along the cell surface. Generally, flagella are long projections. They're going to be very few in number. And cilia are going to have short projections, and they're going to be very numerous. So if you look at the left, here is a flagella, and if you look at the right, this is an example of cilia. So both of these consist of microtubules that are made of the protein tubulin, and microtubules are organized as 9 pairs in a ring plus 2 microtubules in the center, so a 9+2 arrangement. And so this allows the flagella to move in a wave-like manner. And so here you can see that 9+2. There's 9 different pairs surrounding the 2 central microtubules in there. So the cell wall and the glycocalyx, these are important to plant cells as well as animal cells, and these are going to help with cell-to-cell recognition and some other interesting concepts. So the cell wall is mainly found in plants, algae, and fungi. It's going to be made of carbohydrates, mainly cellulose in plants, chitin in fungi, and glucan and mannan are going to be the cell walls you find in yeasts. In the glycocalyx, these are carbohydrates that are bonded to proteins and lipids in the plasma membrane, and we find these in animal cells. So the plasma membrane, or the cytoplasmic membrane, this is going to be similar in structure to that prokaryotic cell membrane that we will study here in the next the next week's material. So it's made out of a phospholipid bilayer. It's integral. It has integral and peripheral proteins, but the differences in the structures are there are sterols, the complex lipids, and there's carbohydrates for attachment and cell-to-cell recognition. And so without these carbohydrates, the cells would not be able to hold on to each other and make long, big sheets. And really, this is one of the reasons why the plasma membrane helps to create tissues. And so, the similar in function to the prokaryotic cells, they are all selectively permeable. So they're going to have simple diffusion, facilitated diffusion, osmosis, and active transport. But the differences here is we can undergo endocytosis. So phagocytosis and a pinocytosis. We can, eat things. We can drink things, or at least our cells can do that. The phagocytosis, the pseudopods extend and engulf the particles. The pinocytosis, that's the membrane drinking. And so there's a lot of these things that the cell wall in a prokaryotic cell will prevent you from doing. The cytoplasm. The cytoplasm is the substance inside the plasma membrane and outside the nucleus. So let me make sure I put membrane in there. And outside the nucleus. The cytosol is the fluid portion of the cytoplasm, and the cytoskeleton is made of microfilaments and intermediate filaments, giving shape and support. The cytoplasmic streaming is the movement of all the cytoplasm throughout a cell. And so cytoplasmic streaming is actually really interesting. And so let's say that you have this cell here, okay, so just pretend that's a cell. You might have this dot, and it might get completely circulated around the entire cell. And this happens a lot of times in plant cells. A lot of times when we look at cells on microscope slides, they're already dead, they're already fixed on the cell, and so we don't see any movement. But if you were to look at something that was alive, like a slice of a plant or something like that, you might see these cells, kind of moving these chloroplasts and other things inside the cell around so that they can get more energy or more light for those particular structures. Though the ribosomes are very interesting. So we mentioned in the prokaryotic section how the ribosomes are a little bit lighter. We have the 80S and the 70S here in the eukaryotic cell, and in the 70S we find those in the chloroplasts and mitochondria. So those would still be susceptible to any type of antimicrobial drug that's going to target those particular cells. The 80S ribosome consists of the large 60S subunit and the small 40S subunit, and these are membrane bound, so these are generally attached to the endoplasmic reticulum, or they can be found in the cytoplasm. So here's a check your understanding question. Make sure you can take a moment to take a look at this. The organelles, so we've already covered a few of the organelles, but we'll cover these other ones in a little bit more detail. So the nucleus, this is a double membrane structure. It's surrounded by the nuclear envelope. It's going to contain the cell's DNA, and that DNA is complexed with a histone protein to form chromatin. And during mitosis and meiosis, the chromatin condenses into chromosomes. And so here you can see a transmission electron micrograph here of the nucleolus here in the middle. That's that dark staining region right there. And then you can see the chromatin all throughout the nucleus. Okay. So the endoplasmic reticulum is a folded transport network. We have two different types. We have the rough endoplasmic reticulum and the smooth endoplasmic reticulum. And the rough ER is going to be studded with ribosomes. These are generally the sites of protein synthesis. And the smooth ER is going to have no ribosomes. It's going to synthesize cell membranes, fats, and hormones. It's also going to be very useful in detoxifying any type of toxin that comes through the body. So here you can see the difference between the rough and the smooth. You can see the rough kind of is textured there on the top picture, and you can see the smooth is not very textured on the bottom. So once again, here's a little bit closer look at that. The Golgi complex, these transport organelles, and they modify proteins from the endoplasmic reticulum. The transports are modified proteins via secretory vesicles to the plasma membranes. And so here, you should know that there's a cis phase and a trans phase, and so there's this is the cis phase, and this is the trans face. And so there's two different sides. So we always have things going in this direction. So it's a very directional transport. The cisterna are these long, kind of flattened, membrane-bound areas that the transfer vesicles are going to kind of go through. And so this comes from the endoplasmic reticulum through the Golgi, and then generally from the Golgi out to the outside of the cell. The mitochondria have a double membrane, and they contain inner folds called cristae and a fluid called the matrix. It's involved in cellular respiration, so ATP production. And some other organelles you might see are the lysosomes, that are vesicles that are formed in the Golgi complex that contain digestive enzymes, and vacuoles. These are cavities that are formed from the Golgi complex. They bring food into cells and provide shape and storage. So here you can see the mitochondria, you can see that inner membrane and the outer membrane there, and so this is very important for production of your of ATP inside your cell. So the chloroplasts are locations of photosynthesis, and these contain flattened membranes called thylakoids that contain chlorophyll. And so these chlorophyll-packed chloroplasts are going to essentially have stacks, and they're going to be in your cells, or not in your cells, in the plant cells and the algae cells. They're going to be in these cells so that they can create that glucose for the cell to burn. So here you can see it in a little bit more detail, and you can see that they are stacks. The thylakoids are these individual stacks, and the granum are these multiple stacks. There is a double membrane, but that it's not really as important as the double membrane that we have in the mitochondria. Peroxisomes, these are going to oxidize fatty acids and destroy hydrogen peroxide. And then the centrosomes are networks of protein fibers and centrioles that are going to form the mitotic spindle and have a critical role in cell division. So here's your check your understanding question. Make sure you guys can understand these questions, and if you have any questions, please ask me. Lastly, let's talk about the evolution of eukaryotes. So life arose as simple organisms between 3.5 to 4 billion years ago. The first eukaryotes evolved about 2.5 billion years ago. And so this brings us to a question, the endosymbiotic theory. So the mitochondria and the chloroplasts both have their own DNA. And so because they both have their own DNA, it's thought that they were brought into cells to do specific functions. And so these larger bacterial cells engulf the smaller bacterial cells, developing the first eukaryotes. And so they ingested the photosynthetic bacteria and became chloroplasts, and then ingested the aerobic bacteria to become the mitochondria. And so here you can see here, and just to note, the mitochondrion came first. So this is first, and the chloroplast came second to the tree of life. So the early cells, the archaea, kind of branched off to it, and then the eukarya decided to envelop their DNA in a membrane, and then take in the mitochondria, and then at some point they took in chloroplasts as well. And so that's the endosymbiosis theory in a nutshell. It goes way deeper than that, but we're going to kind of gloss over it because it's not really that important to what we're studying. So here's your check your understanding question. Please make sure you take a moment to take a look at this, and if you have any questions, please let me know.